Okay, welcome everyone to the Water, Wetlands, and Watershed Seminar for today, September 23rd, 2020. Today we have Dr. Vince Lovko who's going to talk to us about phytoplankton, but first I want to open it up. Are there any announcements either from students in the class or faculty? Do we have any events coming up? I know we have a sort of change in the modality of our events, but Mark, you got any volunteer days out at the seashore?
No, not quite yet. Come October, mid-October through mid-November, we will definitely be having some announcements of volunteer dates to do some planting along the airport road living shoreline, but at present, that no date has been specified.
All right, well, keep your ears out for that. And I know we've already had our monthly meeting for the Wetlands Club for September, but another one coming up in October. We'll announce about that. All right, well then it is my pleasure to introduce Dr. Vince Lovko. He's Senior Scientist and Program Manager for Phytoplankton Ecology at the Mote Marine Laboratory down in Sarasota. He took over this as Program Manager in April of 2013 and was also recently a Project Manager for the Florida Fish and Wildlife Commission, Mote, FWC Mote Cooperative Red Tide Monitoring and Mitigation. So I think we're gonna hear a fair bit about a number of phytoplankton species. I'm not sure where you're gonna exactly where you're going to take us, but Vince has his PhD in marine science from VIMS, the Virginia Institute of Marine Science, College of William and Mary at the College of William and Mary in 2008, and he completed a postdoc at the University of Southern Mississippi. His current research focuses on Florida red tide. You've heard a lot about it in the news. So Karenia brevis or K. brevis, including how to detect and monitor the species and just the fundamental ecology of when, where, why, how they bloom, and ideally how we can prevent or mitigate the impacts of red tide blooms. So with that, Vince, the floor is yours. You can go ahead and share your screen.
Okay, will do. Is everybody seeing an orchid right now?
Yep, we see your beautiful orchid there.
Yes, sir.
All right. Oh, that's not what we wanted to start with. Let me
yeah, you jumped the shark there.
Yeah, a little bit. Okay, let's try that again. Okay.
Perfect, we got it.
All right, sounds good. So first of all, thanks. I'd like to thank the organizers, specifically Christine Angelini for nominating me to be invited to talk. Thanks to David for emceeing the talks. Thanks to the previous presenters that have given me a tough job by raising the bar with their excellent presentations the past 2 weeks. Hopefully I can live up. And thanks to everybody who's listening, both live as well as who might listen later on YouTube. So yeah, I think David pretty much covered me. I've been at Mote for about 10 years and I have a pretty diverse array of projects, but really most of them focus on monitoring of red tide in collaboration with the state. Also, there's a bit of technology development that I'm involved with. We're dabbling in drones and hyperspectral imaging, and then more recently through the state red tide initiative that some of you may heard about, we have a number of mitigation projects. So David mentioned about mitigation. I'm not going to be talking about mitigation today, but feel free to, if you have any questions regarding that or other aspects of red tide, feel free to bring those up when we get to the question period. So I'll kind of jump in a little bit. The changes in community structure in general in the ecosystem can be a really important property for assessing environmental health. We heard pretty thoroughly about ecosystem disturbances last week from the seminar presented by John. He did an excellent job at that. And the overall aim of this talk is to consider changes in the phytoplankton community of the Central West Florida coastal waters with the idea of eventually being able to use that as an indicator for red tide. Also along with that is the idea of seeing if there's any long-term shifts in the phytoplankton community, potentially due to increasing red tides, either severity or frequency. That's not quite what we're going to get into today, but that is our long-term goal. So, to move ahead. So, of course, phytoplankton have a critical significance in the aquatic food web. They form the base of the food web in most aquatic systems. This is a pretty, textbook image of the fixation of carbon and using sunlight by phytoplankton to create biomass and moves its way through through the trophic web with plenty of loss along the way. And that, of course, is very important, but perhaps equally important is the role of the phytoplankton in the chemistry and biogeochemical cycling, and things like the carbon pump. So phytoplankton, of course, are responsible for oxygen production, roughly 50% of the oxygen, assimilation of reactive inorganic substrates, nutrients, in other words, that are then incorporated into particulate and dissolved organic matter that then support the production of pelagic and benthic heterotrophs. Moving along, so I like to show this schematic because it really, when I'm teaching or when I'm giving public seminars, because it really gives a good illustration of how diverse the group that we call phytoplankton are. You know, it's easy to just consider them to all be, you know, kind of the same group and really closely related to each other. And of course they're not. Just as a little bit of a reference, you know, you are here, there we are with all the animals. We're closely related with the cyanoflagellates, and the fungi are not too far from us. But the diversity of the phytoplankton are much broader than that. Here in the heteroconts, we have one of the most common groups of phytoplankton, the diatoms, represented by didylium here. Of course, in the alveolates, we have the dinoflagellates, and that's where Corynnium brevis falls. Way over here with the plants, we have the prasinophytes and the chlorophytes. Then over here, somewhat related to the heteroconts, but not quite, are the cryptophytes and haptophytes. And then way on the other side of the eukaryote and prokaryote line is the bacteria. And trichodesmium is represented here. And this is going to actually, the cyanobacteria are going to kind of play a role in trichodesmium specifically in a bit of my talk today, and might play a really large role, which many of you probably have heard about already, but you know, it's nice to see data that actually supports that. So, of course, as phytoplankton are major primary producers in most aquatic systems, their diversity is going to affect the ecosystem structure and function. And it's also important to note that even within these taxa, there's a great deal of diversity based on terms of size or morphology, ornamentation, the ecology, you know, temperature ranges, benthic versus pelagic, some that exist in the neuston, auto, hetero or osmotrophy, mixotrophy, and so forth. So there's a great deal of diversity, and that leads us to the concept of functional types. So this is the definition from the International Ocean Color Coordinating Group, which basically defines it as that functional groups are groups of species that have a common ecological function. You know, that's implied by the term, I suppose, but this can be in terms of trophic dynamics or in the interactions of the biogeochemical cycle. Some examples include nitrogen fixers. Again, we bring up trichodesmium, calcifiers, DMS or dimethyl sulfide producers, silicifiers, and so forth. And these groupings are not necessarily related to physiological characteristics, but are based on functionality and other characteristics such as cell size, which ties into the trophic dynamics. So this diversity provides a potential redundancy within the phytoplankton community that can provide resistance or resilience in the face of disturbance. And we heard a little bit about resistance and resilience last week. So back to the general topic, can phytoplankton community composition provide an indicator of ecosystem change or disturbance? So I'm going to show a few publications here from other people. So there's numerous examples of the utility of using the phytoplankton community composition as an indicator of ecological change or disturbance. Hans Perl and his group used total phytoplankton biomass along with chemotaxonomic indicators. We're going to revisit chemotaxonomic a little bit later. Basically, this means the chlorophylls and other carotenoid pigments to characterize the response of the phytoplankton community to human-induced, primarily nutrients, as well as climatic disturbances. Mote's own Kelly Dixon did some work with some collaborators a few years back, I think this was 2009, that suggested that certain cyanobacteria species from the Oscillatoria family can serve as a potential indicator of eutrophication. Nothing to do with red tide, just estuarine eutrophication. And then what seems like a lifetime ago now, I chaired a session at SURF that focused on this topic and had a number of excellent presentations focused on the phytoplankton community dynamics in response to natural, well, as much as anything can be natural in this world right now, and anthropogenic changes. And these are, of course, just a few of many numerous examples out there. However, there's not a lot of information, at least not a lot that I'm aware of, the response of phytoplankton community to harmful algal blooms. So that's kind of where some of my interest lies. So of course, we got to visit HABs before we visit red tide. So a little bit about HABs. I'm sure everybody out there knows what HABs are, so I'm not going to belabor it too much. But globally, they're considered to be increasing due to anthropogenic influences in terms of nutrient inputs, warming, other human activities. However, it's important to remember that the individual responses of HAB-forming phytoplankton species are going to vary to different environmental variables. So they're not all going to respond the same. So that it doesn't mean that all HABs are increasing everywhere. Some might decline, some might shift, some might stay the same. We get that a lot when I give public seminars about red tide, and I'm asked to give public seminars about red tide a fair bit. Fortunately, we're in a lull right now, so I'm not asked quite so much. People hear about how HABs are increasing, so they assume that Carenia brevis is increasing as well. And that's not necessarily the case, but that's not where we're going for this talk. So the bottom line is that if there's a coast, there's probably a HAB. Moving on. So of course, that takes us to Carenia brevis, on to Florida red tide. Since dinoflagellate blooms in general are often referred to as red tides, we like to use the moniker Florida red tide to refer to the specific blooms we have here along the west coast of Florida that are caused by blooms of the dinoflagellate Carenia brevis. So Carenia is a neurotoxic dinoflagellate. These blooms often occur annually. They're not necessarily big like what we had in 2018. And the picture that you see currently, and also behind me, it's like I'm surrounded by it, is the red tide from 2018. I had an opportunity to go up in a helicopter and take some pictures, which was my first time in a helicopter, so that was fun, but also nice to see it from that perspective. And you can't quite tell, but if you look really closely, there's a lot of little white dots all over the surface of the water, and those are actually dead fish. In any case, not all blooms are that large. They do tend to occur annually, although they may be small. They can range in spatial extent from a few kilometers to more than 6,000 square kilometers. They can be short-lived, lasting only weeks. They can be longer-lived, lasting more than 18 months. They can be relatively small with maybe tens of thousands of cells per liter, or they can be quite large with tens of millions per liter. And we've actually had cell counts that have broken 100 million cells per liter. That's a lot of cells. The blooms can occur at any time of the year. We do kind of have what I loosely refer to as a season, and that's definitely got blown out of the water in 2018 when we had a bloom that lasted all summer. Generally, we tend to see them first observed at the surface near the coast in late summer to early fall. Initiation is not fully understood, but there is evidence that suggests offshore initiation, presumably triggered by some event such as a nutrient source, with movement in subsurface currents towards shore, where cells collect at density fronts and then are pushed upward towards the surface. Termination is even more poorly understood. One of our scientists here at Mote, Cindy Heil, she's actually got a EcoHab project looking at bloom termination. There's a few fairly well understood factors affecting bloom formation and persistence, and I'll touch on those briefly. One of them has to do with the loop current. As with any phytoplankton bloom, especially in what essentially amounts to a coastal ocean environment, such as what we have here on the West Florida shelf, the stability and movement of the water column is going to be a driving factor. And for Carenia brevis, one of the driving, in the Gulf of Mexico, one of the driving factors is thought to be the loop current. This image is from a paper by Ian Mays et al. In 2015. I believe he was a graduate student at the time. Others have worked on this issue as well, including Larry Brand, Bob Weisberg, and others. So the loop current basically enters in through the Yucatan Straits and penetrates at different amounts of penetration into the Gulf of Mexico, spins around clockwise, and then exits out through the Florida Straits, becomes the Florida current, and eventually meeting up with the Gulf Stream. The progression of the loop current is shown here as sea surface height. The darker the color, or the warmer the color, the higher the sea surface, and that basically gives us an idea of the loop current. So the penetration into the Gulf is thought to be not necessarily a driver, meaning that it'll cause a red tide bloom, but basically that it'll allow a red tide bloom to develop and persist. And generally when that loop current penetrates into the Gulf, it creates conditions partly because of the water moving along this part of the shelf here, kind of sets up the conditions that allow a bloom to persist. You get some upwelling, but the right amount of upwelling that allows those cells to be carried in the currents. When the loop current is lower in the Gulf, then we have a lot more interaction between the deeper parts of the Gulf and the shelf, and then we tend to not have conditions that are favorable to a bloom, or at least to bloom persistence anyways, not necessarily related to bloom initiation. So another likely controlling factor, as is the case with most phytoplankton, is nutrients. This is a paper by a rather large group. Gabe Vargo as the lead author, Cindy Howe is on it, a few people from Mote. Back in, let's see, what is the date on that? I can't quite read it. 2008, I think. I need a bigger screen, apparently. But at this point, roughly a dozen or so potential nutrient sources have been identified that can contribute to the persistence of K. brevis blooms. These include benthic fluxes, land-derived input, atmospheric deposition, dead fish, zooplankton feeding, and others that are described in this schematic here. And of particular note, I wanted to mention, again, cyanobacteria and trichodesmium specifically, that are nitrogen fixers and potentially a contributor to K. brevis blooms, bringing new nitrogen into an offshore system. Okay, so aside from the excessive biomass resulting from an extensive bloom, the primary negative effect from red tides are due to toxins, of course. These are the brevatoxins. We have the brevatoxin 1 and 2 are probably the most prominent ones. They're both intracellular. They're based on two different backbones. This backbone A is what brevatoxin or PBTX1 is based on. Backbone B is brevatoxin 2. Brevatoxin 2, when it's outside of the cell, quickly converts to brevatoxin 3, which is the most common toxin that we see in the water column and therefore in the aerosols. So brevatoxins inhibit the voltage-gated sodium channels in nerve cells. They basically interfere with the ability for it to close, so that it attaches, it permanently opens it, and then you get a flux of sodium ions through the nerve cell, and it overstimulates the nerve cell, resulting in the neurotoxic effects. For humans, even though we're not really here to talk about humans today too much, aside from consumption of contaminated shellfish, the primary route of exposure is through inhalation of toxic aerosols. This figure shows how those aerosols are formed through a process called bubble-mediated transport, where basically very small bubbles rise up through the water. Along the way, they're actually scavenging these lipophilic brevatoxins, where you can get a higher concentration in the resulting droplets. So when they hit the surface, they burst, droplets get shot up into the air, these jet drops and these finer film drops. And those can actually, Rich Pierce, who's here at Mote, he did some work on this back in the day, I think in the early 2000s, where he actually found that you can have 50 times the amount of brevatoxin in these drops than you have in the water. So that's how we get the aerosols. So how long have blooms been around? We think a very long time. In the 1500s and 16th century, there was a Spanish explorer, Alvaro Núñez Cabeza de Vaca, Cabeza de Vaca, easy for you to say. He recorded stories by Native Americans in Florida. He was one of the first explorers to visit the Gulf of Mexico. But these stories of red water and death of birds and fish, and apparently de Vaca's crew, some of them got sick. I think some of them might have died. I'm not entirely sure on that. The information is kind of sparse, but from eating contaminated shellfish. Now, of course, we don't know for sure that that was red tide, but you piece all these things together and it's a good possibility. The written records of red tide don't really start until 1844. And the late 1800s saw a period of frequent blooms that included widespread bird mortalities in 1880. And the first recorded incidents of shellfish poisoning. Now, of course, at that time, they didn't know what was causing it. They just, this poison water was all that they knew. It wasn't until 1948 Actually, it was a bloom that started in 47, but a report was put out in 1948 from the US Department of Interior that described a red tide bloom. Now, at the time, there were no federal or state scientists working on this issue, even though it had been around for a while. So scientists came up from University of Miami. They took samples, sent it to Woods Hole, and that's how that resulted in the identification of the causative organism, which went through a lot of name changes. I won't go into that either, but it's currently called Carenia brevis. So there's another question that we're always asked about every time there's a red tide, or anytime we talk about red tide, is have they been increasing in frequency, spatial extent, or severity? And that's a difficult topic, and not one that I'm going to address here specifically. But this figure is from a paper that does suggest they're increasing, but that's not what this figure is showing. It's really just a conglomerate of interpolated cell counts from 1954 to 2002. So it covers a pretty broad period of time. Now, this paper did make the argument of increasing blooms, but we'll address that shortly. But it does show the region where we tend to see along our West Florida coast where we tend to see blooms. Again, like many things I'll show you, blue means nothing's there. Lighter blue, you get more, you get into yellows and greens and reds, and that's a higher intensity. So this other figure I just brought up was created by a colleague of mine for a proposal that we wrote. That was actually going to look at multiple initiation scenarios for red tide. And this shows K. brevis hotspots by percentage of months where there was Carenia brevis densities above 100,000 cells per liter. And what's really interesting here, so if you recall, the time frame for the other figure was 1954 to 2002, and this is 1998 to 2017. This is an expansion of so this region here is an expansion of this region here, so you can see it a little bit better. What you can see is the primary areas, the hot spots are right around Charlotte Harbor, Sanibel Island, maybe a little bit in the Caloosahatchee, and then up here around Manasota County, the mouth of Tampa Bay, and that's not entirely different than what you see from the Larry Brand figure, which is kind of interesting because they're two very different periods of time. But part of what makes analyses so difficult in answering that question, that long-term question of have blooms gotten worse, is the consistency of data, especially prior to the late 1980s. So this image probably takes a little bit of explanation. So we have dates or time along the x-axis going from 1954 to about 2007. And along the y-axis is actually latitude. So this is down in the Keys, down here at the bottom of it, and the panhandle is up at the top. So what this is showing is where you see black means there was no sample taken. Where you see, and I should also mention that I don't have a reference for this figure because I'm not quite sure where I got it. I probably stole it from somebody else's presentation here at Mote because I really liked it. I have learned how to recreate this myself, and you'll see a more recent version of that later on in my talk. But where there's black, there's no sample. And that's really important to understand. Where there's gray, that means there was a sample, but there was no cells found in the sample. And then again, going from blue through yellow and orange and red are different intensities of cell counts. So, you know, we can see a few things. This area back here actually is kind of interesting. That was in the 1950s, where they were testing copper sulfate for getting rid of blooms. And so there was a lot of monitoring focused around that copper sulfate, those copper sulfate treatments. And then of course, starting in about the mid 90s, there was a lot of federal funding that went to harmful algal bloom research in general. And a lot of that came to Florida for studying red tide. And then that's really only increased over time. And so you can see a pretty good amount of coverage. There's a lot of coverage on the panhandle, a decent amount of coverage in the Keys, especially more recently, and then a lot of coverage along the West Florida shelf. But if you look back in these 30 or so years, it's much more sparse. And what you also notice is that where you do see samples, you see very little gray pixels. So a lot of that sampling was reactionary. There would already be a bloom, and then people would go out and collect samples, typically from shore right along this main area in the central West Florida coastline. All right, moving on. So we do know that red tide can have an impact on other organisms and communities. DuPont and et al. In what is that, 2010, showed massive mortality of epibenthos, but with a quick recovery. This was from the 2005 bloom. Before 2018, the big bloom was one that arguably started in 2004, went through 2005 and into 2006. There was a whole lot of research that came out of that bloom, and I think obviously we're going to see the same thing for the 2018 bloom. But the epibenthos recovered in about 2 years, which sounds like a long time, but it was actually more rapid than was predicted in previous studies. Another one, Flaherty and Landsberg found that red tide associated shifts in the fish communities with species-specific declines due to that same bloom. But these effects were primarily observed in juveniles, which were probably a bit less resistant to the effects of the bloom. Well, adults weren't really affected so much. And even so, these juvenile communities rebounded within a year of the event. So that's great. More recently, a group of us at Mote have been working on stone crab. Most of this have been laboratory studies, and it really all came about because some fishermen had observed that during a red tide that the stone crab were looking lethargic and kind of like symptoms of low dissolved oxygen. So we did a variety of laboratory studies, and basically what we found is that there were effects on adult and larval stone crab, including mortality, behavioral impairment that might impact survival, as well as reproduction and recruitment. And although these were laboratory studies, there was a decline in the crab population following the 2018 bloom, but it's really unclear to know if populations have rebounded, how much the red tide really impacted it. The fishery is in decline anyways, mainly due to overfishing, and the ripping a claw off of a stone crab and then tossing the crab back in is not as sustainable as people like to think. So it's really hard to tell, but this is ongoing work. We've been doing this for several years, so I'm sure we're going to keep doing that. Okay, so now we're going to move on to some data. I'm probably talking a bit faster than I intended to, but that's okay. If we finish a little early, we'll have a lot more time for questions. So the data I'm about to show is somewhat cursory and preliminary, but it's based on 2 different analyses. So we just wrapped up a 5-year cooperative agreement with the state. So we have these ongoing cooperative agreements with the state of Florida to basically execute a lot of their monitoring for red tide and some research, and in the past some mitigation, but now the mitigation has been separated out because there's the state red tide initiative. So we have additional analyses underway, but I did want to show some. I think I was a little ambitious and thought I might have a lot more completed by the time this talk came around. But at this point, we're not going to, as I mentioned before, we're not really going to look at the long-term changes in the phytoplankton community, which would be really interesting to see, especially now that we have a good solid 20 to 25 years of pretty good coverage. But we're going to look at the trends in the phytoplankton community composition during the past 5 years, and then a little bit of a briefer look at about a 19.5 year period of some analyses that were just completed a bit more recently. And interestingly, those 2 patterns are going to be a little bit similar. So first of all, I'm going to describe these stations a little bit. So this is the 5-year data analysis. It covers a period from August 2015 to about May 2020. This was part of our Florida Fish and Wildlife Research Institute and Mote Cooperative Red Tide Program. There's 2 survey types depicted here. I probably could have picked better colors for these dots, but if you can tell, these blue dots are what we refer to as a coastal survey. There's stations that run along the 10-meter isobath, and the pink stations are where we are doing the phytoplankton community composition. All the other stations are just a CTD cast and cell counts for the FWC database. We also have a shelf survey, which are these red stations, and those get a full analysis of parameters. So those parameters, in addition to collecting samples for HPLC analysis of phytopigments and then resulting chemtax analysis to look at the phytoplankton community. We also have a full suite of water column parameters, temperature, salinity, PAR, pH, DO, all those things. And also our chemistry group led by Dr. Emily Hall does nutrient analysis, a pretty large suite of dissolved in particulate nutrients, and then our ecotoxicology group does toxins. And so we're going to look at some of that data. So this is the map I promised you, where I tried to copy that really cool one that goes from 54 to 2007. This one only goes from August of 2015 to May of 2020. And it's a very similar thing. So we have, again, we have the latitude on the y-axis, time on the x-axis, and black means there was no sample taken. We don't go quite as high as the Panhandle, just Tampa Bay, roughly. Black means no sample was taken. This white, which is actually a very light gray, means that sample was taken, but it was the cell abundance was less than 1,000 cells per liter. And then the gradation of yellow through to this dark purple indicates low to high concentrations of Cornea brevis. And what we can see is that we had a couple of different bloom periods. From 15 into 16, 16 into 17, and then our big 14-month bloom that started in late 2017, went all the way through 2018 and actually didn't end until early 2019, although it was much sparser by then. Made it down into the Keys, as we can see here, and into the Marco Island area. Okay, so got some figures here. I'll do my best to describe them slowly. So these are averages over the per survey type over the entire period, average for each year. This is chlorophyll. This is separated by the shelf survey and the coastal survey. Phytoplankton biomass is represented in terms of the total chlorophyll. On the left side of the figure for the Florida shelf, and on the right side of the figure for the coastal survey. Excuse me. The on average, the values on the West Florida shelf seem to reflect the oligotrophic nature of the shelf. The levels at the shelf stations were generally low. There was a peak observed during 2018 due to the large Cornea brevis bloom that was prevalent during the majority of that year. Regional highs were observed during the fall, which you can see in the lower panel. So these are averaged over those 5 years per season. While the chlorophyll levels were generally low during spring, which covers April to May. So, in some coastal systems, you might see a traditional spring high in chlorophyll, but that's not really seen here on the West Florida shelf typically. In contrast to the shelf, the chlorophyll values were significantly higher, in some cases by order of magnitude in the coastal stations. It should be noted that samples for phytoplankton community analysis, which is on the coastal stations, were only collected at those inlet stations that I described before. So just those 4 stations at the inlets. Let's see. So total chlorophyll levels, as I mentioned, were about an order of magnitude higher, particularly during the 2017 and 2018 when they were associated with high K. brevis abundances. Seasonal trends averaged. Seasonal trends average over the 5-year period show the highest phytoplankton biomass during summer months, though the median was higher in the fall for the coastal, and the maxima actually occurred in the winter, as you can see here. Generally, but those levels generally maintained through the fall, falling through the winter and spring back to low levels. And for us, the total chlorophyll, I don't know if anyone out there is a pigment person, but we add the chlorophyll A along with the chlorophyll A allomer and chlorophyll A epimer, which are breakdown products. So all those get added together to give us our total chlorophyll. So in the next figure, this actually describes the phytoplankton variability. So there's a key down here that shows the different taxonomic groups. Did a terrible job with that. And then, so these are absolute, these are relative abundances of the phytoplankton, different phytoplankton groups, totaling to 100%. This is not necessarily reflecting the total amount of chlorophyll. So basically, the major groups that dominated the regional phytoplankton community were the diatom 1 group, or group 1 diatoms, group 1 cyanobacteria, the paradinin containing dinoflagellates. So these are, we call them the group 1 dinoflagellates. These are not K. brevis, and also primnesiophytes. And combined, those groups contributed more than 70% of the total chlorophyll. The phytoplankton community at the shelf stations generally maintained a more stable distribution, with the exception of 2018 and 2019. Major differences were observed among these phytoplankton groups, especially in the cyanobacteria, as well as K. brevis, or the group 2 diatom, or dinos as we call them, and the primnesiophytes in these years. The annual contribution of cyanobacteria increased nearly 10% in 2018, which is the year when there was a large Corynea brevis bloom. Or in 2019, a significant shift was observed in the community composition, which favored Primnesiophytes, which nearly doubled. And there was no major K. brevis bloom. There was a little bit of K. brevis, but it was less than 5% of the total community during that time. Diatoms, both groups 1 and 2, were significantly lower in the shelf in comparison to coastal stations, which makes a lot of sense, and remained relatively consistent across the shelf. Well, at least diatom 1 did. And we tend to see these differences in diatom 1 and diatom 2, and I'm going to get into that a little bit more shortly. But here we saw diatom 1 was relatively consistent in proportions across the shelf, while diatom 2 were usually lower. And cyanophytes, again, the cyanobacteria were dominant groups in the shelf region. So the coastal, so the lower figure here, at the coastal inlet stations, diatoms were the major phytoplankton groups, contributing roughly about 30% of the community, with diatom 1 being the dominant of the group. Similar shifts, as discussed earlier, for the shelf occurred in the coastal during 18 and 19. Massive K. brevis bloom in 2018 was followed by a very insignificant K. brevis bloom in 2019 and appeared to be responsible for the observed changes in the phytoplankton community. In 2019, the community was dominated by diatoms, followed by cyanobacteria, about 30% for each of those, a little over 30 for diatoms, about 28% for the cyanobacteria. Also interesting was the increase in diatom 2, group 2 diatoms at the coastal sites during those periods or years when K. brevis was at low levels or insignificant. Possible explanations for that difference between, and again, we'll talk about it again shortly, but the difference between the diatom 1 and diatom 2 are the different ways that they utilize nutrients. All right, so a bit more. This is probably a bit overwhelming if you didn't like the last figure. But here we show some seasonal variability. So we have the shelf surveys over here, coastal surveys over here. And now these are broken down seasonally by summer, fall, winter, spring, as you can see, and broken down within those seasons by year. So we have the averages per year in each season. Now, in some cases, you see these blank areas like here and here, where we basically did not have that particular survey during that season for that particular year. Sometimes we get blown out by weather and broken down boats and other things that don't allow us to get out. So in any case, similar to before, the same phytoplankton groups shown here, relative abundances totaling to 100%. So one interesting trend we saw from the 5-year data was that the high Corynea brevis during the fall were preceded by high amounts of cyanobacteria, increased amounts of cyanobacteria. And that's pretty interesting. So again here, let's see, where's fall? So we had an increasing amount of cyanobacteria along with the increasing Corynea brevis. In general, this was cyanobacteria 1 or group 1 cyanobacteria, which does include trichodesmium, and they occupied close to 30% of the total chlorophyll. So of course, the explanation that leads to the explanation that K. brevis might have benefited from this growth of trichodesmium and the nitrogen fixing that they brought, especially into the offshore regions in the shelf survey. And also, let's see, as discussed previously, the regional highs of K. brevis occurred during the fall, while in the winter there was more of a contribution from Primnesiophytes and Prasinophytes. Okay, so we also conducted a variety of non-parametric analysis using a program called Primer-E to determine community similarities using Bray-Curtis similarity calculations between samples, and also to identify clusters or groupings based on specific factors or variables that might correlate to the presence of significant abundances of Corynea brevis. So we use multidimensional scaling, which is what these plots show. Ordination of those Bray-Curtis similarities to visualize these similarities or dissimilarities between samples. The taxonomic data was square root transformed to minimize the influence of extremely high or low abundances, which is kind of standard practice for these calculations. We used, so the cluster analysis, so these little groupings that you see represent 75% similarity. So all the samples within that cluster, I should point out that this is actually only for the 2016 and 2017 data. There's a few issues with the 2015-16 data, and we're currently working on the 18 through 20 data. So we're limited to the 2016-2017 data, covering a bloom period that lasted from October 2016. I think I have another figure here. Yeah. So we can see that the bloom started in about October of 2016, seemed to have a little bit of a lull in December, and then picked up again, declining into February and lingering a little bit into March. So what's interesting about that is when we look at the cluster analysis, we do see these clusters that have August and September, and a little bit of others, October, December, but a lot of the October and February samples are in these other clusters that are out here, and this seems to correlate with higher amounts of Corynea brevis as well. So what that kind of shows us, now there's two things to look at. We have these two different clusters. There's one here and one here that both occupy almost the same months. And we did see, using a one-way ANISIM, we did see a very strong correlation, or a very strong grouping by month. That's why I have these listed here. And it kind of looks like, we had a bloom, the community shifted, and then it shifted back. But we do have this little other cluster here that has nearly all of the March samples. But again, if we looked at the abundance chart, we did have some cells or some abundances lingering into March. So the question is, do we have a, sort of a shift back to the original phytoplankton community, or do we kind of have a little bit of a different community resulting? It's probably just a result of the lingering Corynea brevis. Okay, so this shows bubble plots of the abundance of prominent taxa from all of the samples collected during the 2015 to 2017, 2016 to 2017 project year, overlain on that same multidimensional scaling ordination plot. Again, the clusters indicate 75% similarity. And the reason I wanted to show this is because you can see with these bubble plots where the Corynea was, that's the Dino 2. You can see how the group 2 diatoms tend to be present when Corynea is not, and not present when Corynea is present, whereas the group 1 diatoms don't really seem to care. There was a pretty strong positive correlation with the group 1 diatoms. This is a Spearman coefficient, the rho value, and you have a 0.7, which is quite high. Some weaker but still positive correlation with the group 1 cyanobacteria, which again is the trichodesmium, or includes the trichodesmium, and also the a couple of different Primnesiophytes, Primnesiophytes group 5 and 6. So let's see, where are we? So other significant components of the community in terms of contribution to the biomass community structure include the cyano1, as I mentioned, the different Primnesiophytes and Prasinophytes group 1. And again, I pointed out that difference between the diatom group 2 and the diatom group 1. And diatom group 1 contains additional accessory pigments that are thought to serve a photoprotective purpose versus the diatom group 2, which primarily has chlorophyll C and fucoxanthin. So let's see. So we also, this data is not shown, but we use the BioN procedure in primer using Spearman's rank correlation analysis on rank transformed environmental data to try to determine a match between the patterns of community structure and some of the environmental variables. We didn't really get very good correlations in this particular data set. They were less than 0.2 with a p-value of 0.03 when the biomass variables were excluded, which we generally want to do because the biomass variables are going to strongly relate to Corynea brevis, even when as many as 5 variables were included in the analysis. We did see K. brevis abundance alone had a correlation of 0.428, which means Corynea brevis is really driving the community when it's present, especially in high abundance, beyond just the biomass abundance, because we do the normalization to try to take care of that. And also particulate silica had a Spearman correlation of 0.411, which indicates that in the coastal regions, Corynea brevis and diatoms are the primary driving factors for the community composition. So the conclusions for that 5-year study then are that chlorophyll events basically peaked in 2018, both offshore and nearshore regions, and chlorophyll was generally higher in the coastal regions. Seasonally, it peaked in fall in the offshore region, summer in the coastal region, although as I mentioned, the median value was highest in fall with maximal values occurring in winter. The cluster analysis potentially shows recovery of the phytoplankton community following bloom periods, although that certainly needs more research. And the abundance of diatom 1 and to a lesser extent Primnesiophyte 6 and 5, Prasinophyte 1 and Cyano 1, which again is trichodesmium, show moderate positive correlation with Crania brevis. And there also appears to be a difference in the response of different diatom assemblages, the diatom group 1 versus group 2, to the presence of elevated Crania brevis. I think I have enough time to get through a couple more slides. So the 20-year data set, it's actually about 19 and a half. I'm just going to give a very brief synopsis. This work was actually presented by one of my staff, Sumit Chakraborty, at the Ocean Sciences meeting in San Diego back in February. We're working on turning this to adding some more analyses and trying to turn this into a manuscript. The data set covers 8 different projects that involve their own cruises spanning 1998 to 2017. As indicated by the map and the legend, they generally cover that hotspot area that we've identified earlier. So it covers that region pretty well, but if you notice the dates listed here with each of the cruises, there's kind of a lack of temporal continuity between these different surveys. So to jump into some data, so generally speaking, the 20-year trends, this is chlorophyll data. Total chlorophyll is a gray line that's superimposed on Crania brevis counts. They're on the same scale. These are in millions for the Crania brevis counts. And they generally mirror each other a great deal, which again just shows that Crania brevis is driving the biomass when there's intense blooms. And during the last couple of decades, most of the high chlorophyll events either led to, or preceded, or coincided with blooms. The inset here shows the differences between years, with 2007 considerably lower than the other periods, and 2012 through 17 a bit on the higher side. The second graph shows a 20-year average total chlorophyll by month. Seasonally elevated chlorophyll events typically initiated by July. You could see it start to significantly increase, peaking in September and then decreasing but persisting into the early part of the year. So again, these might look familiar now. So the phytoplankton community composition, the studied region, was mainly dominated by diatoms. These use the same, I think we more or less have the same legend that we had before. Dominated by diatoms, cyanobacteria, and chlorophytes, with those groups contributing about 60% of the total biomass. The community dynamic shift during blooms of Crania brevis, much like what we saw in the 5-year data set, with contribution by diatoms and chlorophytes declining while the relative contribution of the cyanobacteria remain a bit more constant. The seasonal distribution of phytoplankton shows a relative contribution of Crania increasing during the fall, followed by a significant dip, and again reoccurring in January. And again, we kind of see that those blooms of Crania brevis tend to be preceded by increases in the cyanobacteria, which we see here. And then the Crania blooms following in September and October. So a little bit, just a little bit more principal component analysis. So here we have the first and second principal components. They do explain about 60% of the total variance in the data, but we don't see any very strong correlations with the majority of the environmental variables. There's some very weak positive correlations with nitrate and silicate. The heat map next to it shows the Spearman correlation coefficients for the phytoplankton community and the environmental data. And again, although about 9 environmental parameters were found to explain about 60% of the variability, there wasn't very much, a lot of variability was not explained. And so it doesn't really allow us to put really a higher emphasis on any of the individual parameters. And so that leads us to conclusions for the 20-year. So the seasonal chlorophyll events typically initiate in July, peak in September, persist through the early part of the year. During 12 of the 19.5-year study, the chlorophyll events preceded or coincided with Crania brevis blooms. Time series plots indicate the blooms were preceded by increases in the cyanophyte and the cyanobacterial and also prochlorophyte abundances. None of the environmental parameters explained about 60%, but that was kind of weak. So a lot of the variability is unexplained. And let's see. So the next steps. So we're like I said, we're working on doing a reanalysis of all this data, trying to bring in some additional parameters, including an upwelling index, winds, currents, and the aeolian dust. And if anyone knows about the iron dust hypothesis, we had a very big dust storm this past year. So far, Karenia is clear. We haven't seen any Karenia at all, really, not even background concentrations. We also want to incorporate more analysis of the taxonomic diversity, both alpha, beta, and gamma diversity, broaden the focus to include the estuarine region. So for a brief period of time back in 2012 through 15, we did only 2 surveys per year into some of the estuaries. We did Tampa Bay, Charlotte Harbor, and went pretty far up the Caloosahatchee. Well, we've returned those surveys to our repertoire, and we're going to be doing those 4 times a year for the next 5 years. And also we want to improve the chemtax ratio matrix by using microscopy and also empirical analysis of pigment ratios from key taxa that we're actually going to isolate from those local waters and grow and look at the empirical pigment ratios. That'll allow us to improve our chemtax ratio matrix, which will give us better results. And nothing's done alone, of course. This is my group, or at least my group as it was this summer. I think 6 of us are regulars, the rest are interns. I tend to get a lot of interns every summer. I had, I think 5 or 6 of them this year. I think my peak so far in the summer was 8. But they really, I don't think Mote would function very well without interns. And a few other acknowledgements, other people here at Mote that contributed to a lot of the data, the chemical ecology program run by Dr. Emily Hall, the ecotoxicology program run by Dr. Pierce, Dr. Heil, who used to be with Fish and Wildlife and is now here at Mote running the Red Tide Institute, and people of the past, Gary Kirkpatrick and Kelly Dixon, of course, many people at FWC and FWRI that are a big part of our collaborative research program with them. And with that, I'm done. Sorry if I got a little long-winded.
No worries.
It's not unusual for me. And sorry if that was a bit overwhelming.
It's a lot of data. Thanks, Vince. We've got some questions from YouTube.
It's probably too much data.
Well, I got a question for you to kick it off, which
Sure.
is the last thing you showed, which is like the PCA and the correlation matrix.
Right. I figured you'd ask about that.
Well, 60% of the variance, I wasn't sure exactly what was the response variable, but 60% was.
Well, it was a combination of 9 variables.
Okay.
So it just basically, it's saying that we need 9 variables to explain even just 60% of the variability. And so that's not very strong.
Is it not though? I mean, we got such a tremendously complex. So I guess the question is that really weak? And if there are, is it that each one of those adds explanatory power but non-significant? Or, from a statistical point of view, why is that weak? If it's a multi.
You're putting me on the spot.
I mean,
no, that's okay. Hey, I'm open. My colleague Sumit, he put those together. I just, when it takes 9 variables to explain 60%, it just I don't know, it just seems like I
mean, I'm an engineer,
kind of throwing in the kitchen sink a little bit.
Sure, I'd like one variable to explain all of it.
Well, not looking at one, of course, and we know when they start to include biomass variables, then that starts to get a bit messy.
Yeah, for sure, right? Because then you're looking at interactions. I would just say that the other thing that people are throwing at this is black box AI machine learning models that explain probably also 60%, but with like 10,000 variables. On the back for doing so. So I'd say that in contrast, I was one of the other questions I was thinking of asking you, whether you had used any machine learning models.
No, we have not yet.
Yeah, I think it's probably coming, and it's exciting but also worrisome. So I'll open it up to anyone else in the room who has questions. One from John Upchurch. This might be a long answer, but what kind of instruments were you using to collect data? So it's probably a variety.
Yeah, so I guess that depends on the type of data you're talking about. We use seabird equipment for. We have a Rosette in SB55. Has a thermal sensor and conductivity and all those kind of things. I don't know if that's what was being referred to. but we use Seabird for a lot of that equipment. For the HPLC, we have a Shimatsu HPLC, high performance liquid chromatography or chromatograph. And then, yeah, so it helped to be a little more specific on the data.
I honestly wasn't too sure. I mean, I guess I was thinking about like counting all the different cells, or like, you know, everything. Like, were you just using a funnel and then like taking it back and extrapolating that, or like how did you exactly know what was where?
Right. So we actually use a Niskin bottle to collect our discrete water samples. Those get brought back to the lab. Well, it depends on which survey we're on. If we're on our larger boat, the Eugenie and Clark, we actually do a lot of the processing on board. But otherwise, we bring it back to the lab. Some of the water gets used for nutrient analysis. We use water for the HPLC analysis, which goes through a filter. It's just standard procedures for that. For cell counts, we actually do those on a microscope. We fix them in a acidic Lugol solution and count them on a microscope.
I can imagine your eyes hurt after that.
Yeah, well, we tend to get pretty high abundances. So background is considered 1,000 per liter. So, you know, that's only 1 cell per milliliter. So, you know, if we count a couple of milliliters, you know, it's not, you know, the error is kind of large when you're talking about that few particles counting. But when we get into bloom situations and you have, you know, tens of thousands and into the millions of cells per liter, it's pretty easy to count by the methods that we use. We use kind of a modified Udemol technique. We use well plates, 24 well plates. We put 2 milliliters in and let it settle. And you can count them in a few minutes, you know, any one sample. So you can get through a lot of samples. We do have a flow cam, but part of the problem is a lot of the samples that we collect, a lot of the data that we generate is for Florida Fish and Wildlife for their red tide database. So they like consistency, and it's been microscope counts for decades and decades. We do have a brand new flow cam that I would like to try to use, but really, if anyone's used a flow cam, I'm not sure it's any quicker than doing microscope counts. Now, if you're trying to do a lot of different species and you have good species files, library files that you can have it count automatically, and because if you know, counting just Carenia brevis is one thing, but if you're trying to count a whole variety of other phytoplankton, then it gets really time consuming to do it via microscopy.
Mm-hmm.
Of course, I could also try to get a flow cytometer, but I don't have one of those yet.
Well, thank you so much. That's really cool.
Yep.
I got a question, David. Vince, thank you very much for the presentation. Lots of information to try to process.
Yeah,
but
sorry
I appreciate
about that.
No, it was great. Appreciate the whole context of the background as well. That's helpful because you hear a lot of different things, but you kind of compiled it in a nice concise manner. My question relates to, is anybody looking at grazers as part of these blooms and what role they may be playing? I mean, obviously a lot of the abiotic drivers of nutrient availability and transport currents and whatnot make total sense, but is anybody kind of looking at the other side that might be suppressing a bloom, or at least managing is a relative term, but either way, regulating the numbers based on just grazing side?
That's an excellent question. Unfortunately, the answer, I don't want to give a hard no, because I don't want to suggest that nobody's looking at that, but it's not a prevalent question that's been asked. Part of it is, I think zooplankton have often been kind of, ignored in the phytoplankton world, especially when we deal with HABs. I don't want to say completely ignored, because there's plenty of work out there that looks at the effect of HAB toxins on grazers, but especially when it comes to Florida red tide, I think that has been an overlooked component.
I mean, it would make sense that the purpose of the toxin is to prevent grazers from eating it. I would think that would be the primary motivator. It's not about us basically being potentially exposed to it or not. So,
right.
But do we know that that's really the case, or like you say, nobody's really digging into that?
To my knowledge, no, we don't. I mean, there's been a lot of, for HAB toxins in general, there's been a lot, some of them have been elucidated. There's the carlotoxin from carlodinium that's thought to basically paralyze its prey because it's mixotrophic. Others have been shown to be allelopathic or to knock down grazers, but I don't think that's really been elucidated for brevatoxins. I don't think anyone's really kind of put the finger on that.
All right, thank you.
All right, any other questions from students in the course? we're a little bit after, so I know people will have to run. If you got to run, please do so. But I have to ask you one thing before we go, Vincent.
Uh-oh.
It is being recorded, but so, this new paper that came out by my colleagues and good friend Jim Jowitz and Miles Medina, and they did a bunch of statistical work to tie terrestrial sources of nitrogen to
Mhm.
red tide magnitude and persistence. And so they concluded that anthropogenic nitrogen runoff did facilitate growth of K. brevis blooms near Charlotte Harbor. And so managing that nitrogen would be a potential solution for that. So I know that's like, obviously we'd love to be able to do something, proactive to prevent these events.
Right.
Do you think that you talk so much about the offshore process. What do you think about the inshore delivery?
Well, yeah. So, I think the general line has been that, they initiate offshore, far enough away from anthropogenic influences that, those aren't going to cause a bloom to initiate. But of course, once blooms are near shore, they can use whatever nitrogen or nutrient sources are around. And that's, of course, true for any phytoplankton. I mean, they are phytoplankton. But, you do get into a competition thing. So like, I think a lot of it depends on the source of the nutrient. So, and the species of the nutrient. So we have seen with Corynea brevis that, and I think this is pretty well known, that it's pretty good at using some organic forms of nitrogen, such as urea, amino acids, and some other things. And obviously it can use inorganic forms as well, but might get outcompeted by other phytoplankton. So like one of the things that we've done in our lab, doing various challenges studies is if I go out to our dock right out here outside of Mote and take a sample and add even just a little bit of it to Corynea brevis is going to go away and diatoms are going to go crazy because the few diatoms that are going to be in that sample they're going to grab all those nutrients and they're going to way outcompete Corynea brevis. So, if studies are done that's taking doing a challenge of estuarine water or whatever source they're using against Corynea brevis itself in culture, that's not really a fair fight. Now I'm not, to be honest, I'd have to take a look at that paper. I'm not I don't think I've come across that one yet.
Yeah, I've only read the abstract, so to be fair.
Yeah. But on the other hand, though, I mean, it's definitely a question that needs to be asked, and I think that's becoming more understood. So, I don't want to get anyone in trouble, but we had that 3 years of doing the estuarine surveys. We're only doing them twice a year, once in the wet season, once in the dry season. And we did that over a 3-year period. So it's only 6 surveys, pretty spread apart. But then those were ended because it wasn't thought to really be pertinent to the program, which is a red tide program. So now we're bringing them back and we're going to do them quarterly for 5 years. So I think that's going to give us a lot more information because I do think that's something that's been a bit overlooked. And it's not so much, am I saying that I think it definitely is a driver in persistence of Corynea blooms? I mean, it may or may not be. I'm not saying it's not, but I'm not saying it is. But how do we know if we don't look at it?
Yeah, for sure. All right, well, Vince, thank you so much for coming to us virtually today. And thanks, students, just all remember it's a 1 page or less write-up just every week. And by the end of the semester, I think Mark and I are both quite flexible about that. And with that, thanks, Vince. We'll talk to you soon. And
all right.
Have a great day.
Thank you. I appreciate it. Take care.
Oh, yes. Give your emoji response, or you're welcome to unmute and clap. As well. But all right, thanks, Vince. Talk to you
Thanks.
soon. Cheers.
Yep. Bye-bye.
Bye.